Good morning, students. Today our discussion topic is muscles. In this topic, we will study about different types of muscles and how muscles contract. We have three types of muscles found in our body. One type is known as striated muscles, also known as skeletal muscles, as they are connected with the bones. Second type is cardiac muscle, and the third type is smooth muscles. Striated muscles, they appear like stripes, means they show regular striations at intervals. Cell structure is multinucleate syncytium, and shape of the cell appears to be long unbranched cylinders. Within these unbranched cylinders, contractile proteins like actin and myosin filaments are arranged into parallel bundles of myofibrils. As I have already told you that they are distributed over the skeleton, so that's why we call them skeletal muscles. They are neurogenic in function, means they can be instructed by the nervous system to start contracting. So they cannot contract and relax on their own. Rather, they are instructed by the nervous system to start contraction. Second is cardiac muscles. They are actually making up the heart. They also show strips at regular intervals. And unlike skeletal muscles, they are uninucleate cells, but they are joined by intercalated discs. These cells are shorter with branches that connect to adjacent cells. They do not show long fiber-like appearance, just like striated muscles. But like striated muscles, they are also having their contractile proteins organized into parallel bundles of myofibrils. Heart is the organ that is consisting of these muscles, but these muscles are myogenic in function. They can contract and relax on their own. They do not need nervous instruction for their contraction. But in order to maintain or regulate the pace of the heartbeat, so these muscles can be controlled neurogenically by the brain. The third type is smooth muscles. They do not show any striation. They are consisting of uninucleate cells. Cells are long, unbranched cells that taper at either end. Contractile proteins are also not organized into myofibrils, and these are normally found in the tubular structures, just like arteries, veins, arterioles, trachea, bronchi, esophagus, stomach, a small intestine, large intestine, fallopian tube, or uterus. And like skeletal muscles, these are also neurogenic, means they need nervous instructions to start contracting. Now, this is just a general outline of the striated muscles, that is the skeletal muscles, and the muscles shown here is just a segment of the bicep. This is a short length of the muscle fiber seen here. And you can see that contractile proteins are arranged specifically into myofibrils, and they are showing parallel, regular interval striations. And this is a single nucleus shown in the short length. You can see here. Remember one thing, that for muscle fibers, we need to discuss it with different terminologies, like a cell membrane that is enclosing the muscle fiber is known as sarcolemma. Cytoplasm found inside the muscle fiber is known as sarcoplasm. Endoplasmic reticulum found in the muscle fiber is known as sarcoplasmic reticulum. There is a network of transfer system of tubules that start on the sarcolemma, and means they are connecting with the sarcoplasmic reticulum inside. Contractile proteins are in the form of myofibril bundles, and a large number of mitochondria are also found around the myofibrils. Here we go with another detail of the structure of the muscle fiber. You can see the short length, sarcolemma, sarcomere, and myofibril. Here you can see that these are the T tubules that are making a network inside, and these T tubules are further connecting with this sarcoplasmic reticulum. This is actually the bundle of myofibrils here, and these are the mitochondria clustered around myofibrils here. This is you can see here the electron micrograph of the muscle fibers that has been enlarged 27,000 times, and here you can see the myofibril. And these are the appearances of mitochondria, and this is a segment of the sarcoplasmic reticulum shown. Here we go for the actin and myosin filaments, how they are found. So thick filaments that are shown in the myofibrils, these are known as myosin filaments, and thin filaments shown here are known as actin filaments. So means you can say these are actin proteins and these are myosin proteins. Okay, here you can see that these thick dark brown lines, these are representing myosin filaments, and these light brown thin lines are shown here. These are known as actin filaments. Okay, this is A band that covers the area where the myosin filaments are existing. This I band is the region where unoverlapped part of the actin filaments exist, and this is the H band that shows the unoverlapped part of myosin filaments. So this is the overlap part where myosin filaments and actin filaments are overlapping each other. This sarcomere is actually a segment between two consecutive Z lines. So what is a Z line? Z line is a disc-shaped structure that connects all the actin filaments in that myofibril. And this M line is a disc-shaped structure that connects all the myosin filaments present in that sarcomeric region in the myofibril. Whereas the distance between, or you can say segment between two consecutive Z line is known as sarcomere. So whenever the contraction does take place, sarcomere is actually reducing in its length, or you can say contraction is occurring within each sarcomere. So in one myofibril, there are thousands of sarcomeres existing parallel to each other and running one after each other. Here you can see that this is the myosin filaments, thick myosin filaments, and these are the thin actin filaments. This is the M line connecting these myosin filaments together, and you can see very small heads of the myosin filaments. These are known as the myosin heads. They are likely to interact with the actin filaments. In this slide, we are going to study about the sliding filament model of the muscle contraction. If you just have an enlarged view of the actin filament, what you see here, that actin filament is consisting of tropomyosin, actin, and troponin. In a state when the muscles are not contracting, tropomyosin and troponin have blocked the actin molecules, so they are not exposed. Okay. This is the myosin head, and this myosin head that is projecting above the myosin filaments. So these myosin heads are able to interact with actin, but in the non-contracted state, myosin heads are not interacting with actin molecules because they do not have any excess. Okay, but when contraction does begin, so these troponin and tropomyosin change their shape and they skip away and they expose the actin molecule. So that makes this myosin head to attach with the actin molecule. When myosin heads attach with the actin molecules on the actin filaments, so this causes a little distortion and tilting in the myosin head. So they tilt inward from both sides, from this side as well and from this side, and upper and lower myosin heads, they are tilting inward. So when they are tilting, they are stretching actin filaments inside the sarcomere, inside towards the M line. So this will result in the reduction in the length of the actin filaments and overall reduction in the length of the sarcomere, right? So this is how contraction does take place. But remember one thing, that once the myosin head is attached with the actin molecule and it is tilted inward, reducing its length means shortening the sarcomere. So the condition is reversed with the help of ATP. There is an enzyme attached with the myosin filament, and this enzyme is known as ATPase. that hydrolyzes ATP, releasing phosphate. This phosphate attaches with the myosin head, causing a distortion, and this distortion makes this myosin head detach away from the actin molecule. When it detaches away from the actin molecule, so actin filament is reversed to its normal position again. Sarcomere is resuming its normal status again. Okay, it gains its length back. Okay, but for continuous muscle contraction, so these myosin heads need to get attached with the actin molecules again and again in the repeated activity. But how this muscle contraction is induced in the muscle fibers or the skeletal muscle fibers, particularly instructed by the nervous system. So what we see here, that at the neuromuscular junction, the motor nerve fibers at the neuromuscular junction, they release acetylcholine by exocytosis process. So when the nerve impulse reaches the dendrite end of the motor neurons at the neuromuscular junction, that depolarizes the synaptic membrane. So at that region, and acetylcholine vesicles merge with the membrane at the synaptic knob and release acetylcholine molecules by exocytosis process. These acetylcholine molecules interact with the receptors on the sarcolemma of the muscle fiber. That makes the sodium ion channels open, and sodium ion channels start moving inside the sarcolemma, causing the membrane becoming depolarized. This depolarization of the sarcolemma makes to generate an action potential, but that action potential is carried by the T tubules, and these T tubules carry the action potential inside the sarcoplasm towards the sarcoplasmic reticulum. So this action potential is transferred to the sarcoplasmic reticulum. Membranes of the sarcoplasmic reticulum get depolarized. Here within the sarcoplasmic reticulum, there is a huge cluster of calcium ion that has already been settled here. So these calcium ions diffuse out of the sarcoplasmic reticulum as calcium ion voltage gated channels open. So these calcium ion voltage gated channels, as they open, calcium ion diffuse out, and these calcium ion bind with troponin molecule. And this binding will result in distortion in the troponin and later in the tropomyosin. They change their shapes and they expose actin molecules towards myosin heads. Means they just skip away exposing actin molecules. The moment actin molecules are exposed, myosin heads attach with them. So detachment of myosin head is due to ATP hydrolysis that releases phosphate. Phosphate attaches with this myosin head, causing a distortion, and that detaches away from the actin molecule. But for continuous contraction, again and again, myosin head needs to interact with the actin molecules. But once the muscle contraction is not required, no more acetylcholine is released from the motor dendrites. So no more depolarization of the sarcolemma. So no more action potential or depolarization of the sarcoplasmic reticulum. So means no more calcium ions are diffusing out. Rather now, so calcium ion pumps. These are the ATP powered calcium ion pumps. They are activated. These are located in the membranes of the sarcoplasmic reticulums, and these pumps collect calcium ions from the sarcoplasm and actively pump into the tubes of the sarcoplasmic reticulum to build a concentration gradient so that these calcium ions can be used again. So this was about the muscle contraction, how this muscle contraction is induced by the motor neurons when it carries instructions from the central nervous system. And this was all about the muscles, its types, and its muscle contraction. I hope you would have learned a better understanding. Thank you very much.